like Arvin said, I'm a PhD student in the Mathematics Institute in Oxford, and I'm going to talk about my some work that I did in my PhD on non-equilibrium physics of brain network dynamics. But the focus is not so much on the results. I also want to try and take you through like why this is an interesting way to look at the brain, and also like go through some of the mathematical foundations that allow you to apply this to data, and then also some practical techniques that you can apply to your own data. Okay, so this is the structure. I'm going to start with kind of an intro to statistical mechanics and why a shift from equilibrium to non-equilibrium statistical mechanics is even relevant. What are the equilibrium assumptions we're currently making in models of the brain? Then I'm going to kind of go through a mathematical introduction to non-equilibrium steady states, some of which we've already seen earlier. A little bit about some of the results we already have, some practical techniques for your own data, and then a bit of an outlook for what we can do next. So, yeah, so the perspective with which I kind of study the brain and other complex systems is this idea that it encodes information through spatiotemporal patterns of activity, and this is distributed over a complex network. So we're neither in the kind of regular setup of a statistical, for some statistical mechanics models with like lattices, neither are we modeling a gas or something that's completely disordered. We're in this middle ground, which has lots of challenges, but is also produces really interesting emergent behavior. And so, yeah, there's a lot of frameworks from statistical mechanics and nonlinear dynamics that can be used to study systems that look like this. So in particular, neural systems mirror a lot of the stuff that people have already studied in statistical mechanics. So for example, if you look at just oscillations in the brain, you're in the framework of coupled oscillators or coupled spins, which have been studied extensively in statistical mechanics, and you can use things like the Kuromoto or the XY model. If you're at a different scale, so if you're looking at neural spike trains and you turn these into like binary, you can turn this into kind of binary data, which looks a lot like Ising models or spin models. And then when you have noisy continuous valued recording, so things like electrophysiology or neuroimaging, then you're in the domain of basically nonlinear stochastic dynamics. And there's lots of techniques that we can use to analyze this kind of data from this perspective. Okay. So for some of you, this will be totally new, and for some of you, this will be super basic. But the statistical mechanics picture kind of starts with equilibrium statistical mechanics, also known as statics. And so what you have is very large systems that have lots of microstates, for example, the positions of individual molecules, and then you have a set of macrostates. So these kind of ensemble statistics. So an example would be, let's say you flip two coins, and here are the different configurations you can get. And if you look at the macrostates, there's a different number of microstates that correspond to that macrostate. So here there's only 1 and 1, but here there are 2 possible micro configurations that lead to that macro configurations. And this is where the kind of definition of entropy comes from. So this is from Boltzmann and then later formalized by Gibbs. At thermodynamic equilibrium, all your microstates are equally probable. And so basically the entropy, the level of disorder, is just the number of microstates logged that have the same macroscopic properties, right? So just how many possible configurations could we possibly be looking at? And this is also why it's a measure of uncertainty, because you don't really know what the microstate is, depending on how many possible ones there are. Okay. Gibbs generalizes further and said, okay, we're not just interested in isolated systems. What if they can exchange energy? Now your energy can fluctuate. And what you have is some energy function defined on your microscopic configuration, which can really be anything. It means the positions of or the values of everything in your system. And they show, or the kind of this formal, the theory, the key theory of statistical mechanics, equilibrium statistical mechanics, is that this is the distribution of microstates. And so it's called the canonical ensemble or the Boltzmann-Gibbs distribution. It basically tells you how often should we see each configuration based on its energy alone. So yeah, all that matters is the temperature and the energy of the configuration. Okay. So this on its own has no dynamics. It's just a distribution. And what this basically means is that you're just trying to take samples from this distribution. Any fluctuations is just purely noise. There's no actual kind of dynamics in the system. And so we just use Monte Carlo methods to try and draw samples from this distribution. Okay, and then this is the entropy that you might be familiar with, which also coincides with Shannon's entropy for a distribution in general. Okay, and then Jaynes came along in 1957 and produced these 2 papers that basically linked statistical mechanics with information theory in this really elegant way. And what he basically said is that these energy functions for more general systems don't have to just be seen as physical energy, but can just be seen as constraints on your system. Now, what this means is that the Boltzmann-Gibbs distribution is the distribution that maximizes the entropy given these constraints. So what that means is if we want to find the distribution that matches our data but makes no other assumptions, this is the best distribution to take. And it also allows you to use lots of the theory that had already been developed in statistical mechanics to now model data science. To now model data and like develop techniques in data science. Right, so maybe the example that is most familiar is the Ising model, which obviously has implications for neuroscience because it's the maximum entropy distribution for binary neurons where you have constraints on the mean activation rates and on the correlations. So if you have some data and you measure how often each neuron is spiking and you measure the pairwise correlations between them, it's the model that makes the fewest assumptions about everything else is the Ising model. And then once you fit the Ising model to your data, you can do things like look for criticality, just look at the connectivity structure that you've inferred, etc. And I'm going to go through one approach or one and a half approaches basically on how you can fit this to data later on. But this is old work. I mean, this is there's a really good book chapter by John Hertz, who I think is an Audita person, on how you can fit this in all different places. So non-equilibrium, equilibrium, etc. Okay. So non-equilibrium statistical mechanics is different. So systems are not at thermodynamic equilibrium, and now they're dissipating energy to their environment. And at the microscopic level, you have these fluctuations that are large enough that the system behaves kind of probabilistically. So we're not interested in the thermodynamics, say, of like a large liquid. We're talking about things like cells, neural activity. Quantum mechanics is also described by these kind of fluctuations. And at the physical level, what you have is dissipation of heat, right? So a system is basically dissipating heat to its environment. And so what happens here is that basically by producing entropy, the dynamics become time irreversible, whereas the equilibrium systems were time reversible. This basically means that if you watch a movie of the system, you won't be able to tell the difference between forwards and backwards for an equilibrium system. For example, something that's just purely noisy, but you can differentiate it for a system that's out of equilibrium. And this is linked to the idea of the second law of thermodynamics that entropy can only go one direction in time, it can only increase. So if you flip it, you could see things that aren't possible. So for example, imagine you see a glass explode. You would never see the glass go back together again because that would be a decrease in entropy, which we know is impossible. Okay. So these systems do not admit a simple energy function. You can't just describe their distribution as you can in the equilibrium state, and they're time dependent even when they're stationary. So all the things I'm going to talk about today are things that are in a steady state, so the distribution doesn't change, but non-equilibrium systems are still time dependent. Okay. So here's kind of a summary. On equilibrium, the systems are defined by an energy function, whereas non-equilibrium ones are defined by their dynamics. Equilibrium systems always have a Boltzmann steady state, or depending on, or something that is just a rotated Boltzmann distribution. In non-equilibrium, it's arbitrary. Okay, we'll get to this, but usually equilibrium systems are defined by symmetric interactions, which is one of the reasons why this is interesting for neuroscience. When you're using equilibrium statistical mechanics, you're usually assuming that things are coupled reciprocally. And if this is not the case, then you usually find yourself in this perspective. So maximum entropy is an equilibrium paradigm, and it's been used a lot to analyze neural data. Maximum caliber is a non-equilibrium extension, which is somewhat more complicated. These are time reversible and therefore have no entropy production. And these are time irreversible and have a positive level of entropy production. Okay, so now we're going to move on to non-equilibrium steady states and the mathematics behind them. So hopefully everyone's still with me. Okay, so I'll start in continuous state spaces. So here we're typically in the realm of stochastic differential equations, which have two parts. So you have some state. This can be a vector, so it can be, say, the activity in a bunch of neurons, or it could be something that's moving around in XY space. And it's composed of two parts. You have the drift, which is the forces that are actually pushing this thing around, and then you have the diffusion, which is the random noise. And it's basically governed by a Wiener process. And then you put these two things together to give you the diffusion matrix. Okay. So, for example, you can see here, this is a limit cycle with additive noise. So I'm basically going around the circle, but I'm not doing it perfectly because there's diffusion. Okay, so every time you simulate a trajectory from this system, it's different, which makes it difficult to model. But what you can do instead is say, I'm not interested in the exact value of x of t. I'm interested in the probability that a process, if I simulate an infinite number of these, takes value x at time t. And then that gives you what's called density dynamics, which are given by this Fokker-Planck equation, which looks a bit complicated, but it's a partial differential equation that tells you how p of x of t changes in time. So for this limit cycle, for example, if I start with some uniform distribution, it's basically going to close down to this donut. So you can imagine a limit cycle, circle plus noise, looks like a donut. Okay. And it's given by the divergence of this, which we call the probability flux. Okay, so we're interested in steady state. So this is situations where this probability distribution is no longer moving. And that occurs when it's no longer changing in time. So when this is 0, which means that your flux has got to be divergence free. So that's a mathematical thing that basically means that this operation on it is something to do with its derivatives is 0 everywhere. Okay, yeah, so your steady state is when this happens. And for some processes, this means that you're going to tend towards it, right? So then you have a stationary distribution that doesn't depend on time. So you can see here, it's actually going to look like a donut. Okay. And so then we have this steady state flux, so which no longer depends on time as well, is divergence free. And this is what separates equilibrium from non-equilibrium systems. So this as a condition is weaker than saying that this is 0 everywhere. So if this is 0 everywhere, then its divergence is definitely 0. But it can be non-zero everywhere and have divergence 0. And this is what separates equilibrium steady states from non-equilibrium steady states. So when this is 0 everywhere, you're in equilibrium. And when this is a non-zero anywhere, then you're out of equilibrium. And you end up with something that looks like this. So you have what's called a net probability flux. So even though your trajectories stay on the donut with the right probability, you can see they're moving with, this is 2 trajectories, they're moving with some net direction around the circle. They're following this probability flux. This means that it's time irreversible. If I showed you this moving backwards, they would move the other way around the circle. And so you can differentiate between the two. Okay. And so this very nice clean quantity that measures the distance from equilibrium, which is also known as the level of irreversibility, is the entropy production rate. In physical systems, this actually tells you how much heat is being dissipated to the environment. In neural systems, as we'll get to, it's more of an abstract statistical quantity that tells you how far from equilibrium you are. You can see that obviously if S is 0 everywhere, then this is 0. And you can believe me if you can't see it clearly that it's positive otherwise. Okay. So this is a really nice clean quantity that people have tried to compute for neural data. All right, one more mathematical tool that we'll need is the Helmholtz-Hodge decomposition, which tells you that any non-equilibrium steady state can be split into 2 parts, reversible dynamics and irreversible dynamics. Okay, so again, we'll use this example of the noisy limit cycle. We're going around the limit cycle with noise. The reversible part just keeps you on the stationary distribution. It's just related to the gradient of the probability distribution. And then the irreversible part is the bit that sends you around the circle. And if you can split this processes up in 2, you can really see what's going on, right? So you have these forces which are kind of keeping you on the donut and these that are sending you around the donut. Okay. So here's an example, one that we'll come back to with reference to neural data, is the Ornstein-Uhlenbeck process. So this is just a linear SD. Okay, so you have just a drift matrix and a diffusion matrix. Okay. When the eigenvalues of B have the right property, then this is going to decay to a mean 0 Gaussian steady state. And you can work out the covariance of this from the Lyapunov equation. And we've known for a long time that this is reversible if and only if this holds. It's quite easy to show. And then if you have isotropic diffusion in your system, so you're adding noise to each region or each node individually, then it's reversible if and only if the drift matrix is symmetric. So this comes back to what I showed you at the beginning, that symmetry means equilibrium typically, and asymmetry means non-equilibrium. Okay. Another example, also somewhat related to neuroscience, is the Kuromoto model. So this is a limit cycle oscillator where you just kind of go around the circle and they're coupled and they can go in phase and out of phase. We know that it's reversible if there's no internal frequency. So if you have coupled oscillators with symmetric connections and they're not doing their own oscillations, then we're in an equilibrium steady state. Even if you just have one oscillator on its own, if it goes around on its own, oscillatory, then it's irreversible and it's out of equilibrium. So if you're looking at oscillations, you're always kind of naturally in the non-equilibrium paradigm. And again, we can see that symmetry is required for this to be in equilibrium. All right, so the next bit of maths, and probably the last bit of like very intense maths, is on discrete state spaces. So people have also modeled brain dynamics with discrete states, with things like hidden Markov models. So in discrete states, we have a different paradigm. We're modeling this with what's called a master equation, which tells you the probability of being in each discrete state at a time t. And so you can imagine this as basically jumping between discrete states, like a walk over a graph. And what happens is you have this transition rate from j to I, which tells you with what rate you're jumping between nodes. And the word rate is a bit vague, but there's basically a slightly more mathematical explanation for what that means. And you can see again, say I'm jumping between these nodes, I have a probability distribution that's eventually going to settle down to something that looks basically constant in time. So again, when is this going to be steady state? When the probability distribution no longer changes in time, which means that when that is zero. That is zero when your steady state distribution solves this very simple equation. And so, for example, this is the steady state distribution for the walk I was just showing you. And we have exactly the same quantities now in the discrete case. So you have the discrete flux. When this is zero, you're in equilibrium. And when this is non-zero, you're out of equilibrium. And this gives you something that's kind of well known for anyone who's done stochastic processes, the detailed balance condition, which tells you if your discrete walk process is reversible or not. And you have a discrete form for the entropy reduction. So the same quantity that I showed you in the SDs, but in the discrete case. Again, it's zero if this is zero, so if the detailed balance condition holds, and it's positive otherwise. Okay. So here's an example. Again, if you just take a random walk on a graph, so you take a random network or some particular network, and you jump between nodes based on the probability, if the network is undirected, then it has reversible dynamics. If the network is directed, it's typically out of equilibrium, not always. It's not a perfect condition. You can construct examples where it's still in equilibrium, but if it's undirected, it's always reversible. So again, reversibility is related to symmetry between interactions. Okay. And one more example. So if you take the kineticizing model, so this is the one that is basically has dynamics rather than just having a steady state distribution you take samples from. This is an equilibrium, again, if and only if it's symmetric. And then it will converge to the distribution I showed you earlier, the equilibrium one. If it's asymmetric, we don't have a closed form for the stationary distribution. And so you have these dynamics, but you can't really say anything about what they're going to look like when it's stationary, when it's run for a long time. Okay. So now I'm going to show you some of the results quite quickly that people had already found trying to apply non-equilibrium physics to brain dynamics, including some results of our own. And then I'll show you some simple practical techniques for relating the theory that I just showed you back to neural data. Okay, so the story kind of begins with this paper in 2021 from Chris Lin in Danny Bassett's group. That they used a very simple approach that I'll show you in a second to basically show that the level of entropy production was higher in tasks compared to resting state. And this is using fMRI data. And they showed that the level of entropy production was significant. So if you shuffle the data and you do the same process, it's much lower. And that you see this kind of increase in entropy production in tasks. They also looked at like level of cognitive exertion. So if you look at working memory in the fMRI, you have tasks that are difficult levels of difficulty. And they found that more difficult tasks had high entropy production than easier tasks. So given this finding, lots of people tried different data sets. So for example, there's this paper that shows that it decreases if people go to sleep. It decreases if you have Alzheimer's disease. It decreases if you suffer from diseases of consciousness. So if you're in a minimally conscious state or an unresponsive wakefulness state, it decreases significantly. And it decreases if you take LSD. So that's just some examples of how this quantity basically varies between conditions. And people came up with lots of different levels to ways to compute this. So you can focus on irreversibility, or you can focus on the actual entropy production quantity in a purely statistical way or in a model-driven way. So you assume a dynamics for the system. this is kind of an abstract quantity that we don't really know what it means, right? This is a quantity that in physical systems measures heat dissipation. But at this kind of coarse-grained level of neural data, we're not talking about heat dissipation. We're not talking about energetics at all. We're talking about kind of a statistical correlation that we need to kind of understand where it comes from. Okay, so that's at the neuroimaging scale. At the neuronal scale, people also did this. So there was a couple of papers. This one that kind of came up with a more complicated decomposition, but I'm not going to focus on that. I wanted to focus on this idea of where does irreversibility come from at the neuronal level. So they took salamanders who were watching natural movies, which are irreversible, and were watching Brownian movies, which are reversible. And they wanted to see if the stimulus, the irreversibility of the stimulus, is what's causing irreversible brain dynamics. And what they found is that it's not, that they showed the opposite, really, that there was higher irreversibility for Brownian dynamics than natural movies. So even though we know that somehow what you're watching is being represented in the neurons, the irreversibility didn't match up. And so it's coming from some other source. There was slightly more recent work from this year, actually, that showed that actually this is, they got an opposite result. So Gabor filters were lower than passive movies, which were lower than active movies, so natural movies. So it's still an open question of in neuronal cells where you should know to a certain degree what's being represented, we don't know how the stimulus irreversibility affects the actual dynamics irreversibility. Okay. One thing that most people are in agreement on is that we're not measuring heat dissipation. It's not about energetics at all. All right. So you might be wondering that this is actually quite far from neuroscience. This is very much statistical physics. But there are people who are much more neuroscientific than I am, who've basically been doing this without saying they're doing this. So, for example, there's two labs, these two papers from Oxford, from people who look at hidden Markov models for modeling neuroimaging data. And they were looking at asymmetry in transition rates, both in this older paper from 2017 and quite recently, where they're looking at the cycles with asymmetric transition rates. And effectively, this is doing the same thing. You're looking at irreversibility and broken detail balance without using words from statistical physics. And so this is something that, that maybe more hard-headed neuroimaging people are interested in, but don't necessarily use the same language that I'm using. Okay. So now I'll go through a couple of practical techniques, which include some of the results that I came up with in my PhD, and then I'll talk a bit more about where this can go. So this original paper in 2021 used a very, very simple method, and it's actually quite coarse. So they wanted to build a Markov chain, either in discrete or continuous time, with discrete states from neuroimaging data. And so all they did was take their data, treat it as vectors in a high-dimensional space. So this is 100 regions in fMRI data, and just cluster it with k-means clustering. This gives you basically discrete states, depending on which cluster the activity is in at that time. And then all you have to do is infer the probabilities of transitioning between clusters. And this kind of takes advantage of the fact that inferring a Markov chain from data is very, very easy. You just count the numbers of jumps between cells, right? You want to figure out what's the probability of going from one to another, where you just count how many times you went from J to I and divide by the number of times you went from J to something. So using this very simple thing, you can basically calculate transition rates and measure the entropy production. Okay, so this is very simple. It's something you can do really in minutes for your data set, but it has some caveats. So this is something that people have been interested in for a long time, and it definitely matters when you talk about neural dynamics, is that coarse graining has really complicated effects on irreversibility. So if you obscure anything, so if you approximate the continuous process with discrete states, which is what we're doing with this method, if you observe things coarsely in time, so you don't see all the time points, you just see snapshots, or if you only see some of the variables, so there's some variables you don't see, all of this gives you a lower bound, a theoretical lower bound on the true irreversibility. Basically, you don't have enough information to figure out the true irreversibility. And in general, it's a pretty bad approximation of the entropy production rate. So in this non-neuroscientific paper, we wanted to see basically how bad is this, how good is this? And we showed that in the limit, if your cells get really infinitely small, it converges, but in practice, it's really terrible. So, this is the entropy production rate. The true entropy production rate is 25. And as you increase the length of your time series, you're getting closer to 5. So you're still like miles off the true entropy production rate. And this was in 2D with lots of states. So they were in 100D with 8 states. So you're really coarsely approximating it. The other thing that's very difficult is when you coarse grain a process that is Markovian, so it has no memory, you induce memory in the process. So if you're approximating this with a jump process between cells, but it's actually underlying some continuous process that looks like this, it's no longer Markovian. You now have a memory, right? So if you've just entered one cell, it's more likely you're going to jump back than it is that you're going to jump forward to a new cell. And so this introduces all kinds of problems and errors in your entropy production rate estimate. Okay. Yeah, another really simple method, something you can apply in minutes, was developed by Gustavo Decco's lab, which they call inside out. And they basically look at time lag correlations. And we've seen some talks about time lag correlations, but this is one thing you can kind of use them for, is to measure the level of irreversibility. So you measure the correlation of one variable with a forward lag, and then you measure it with a backward lag. And if this is asymmetric, then you'd say there's time irreversibility. And if it's symmetric, you say there isn't. And so using this, you can show that you have higher irreversibility awake versus asleep, or awake versus anesthesia in propofol. And it's quite easy and something you can really just run simply. And it has a good intuition because it gives you a directed measure of connectivity, unlike functional connectivity. So this is one of the reasons we're interested in these kind of techniques, is because often we're making symmetric or equilibrium assumptions without knowing. So if you measure functional connectivity, you take the correlations between all of your regions, that's symmetric intrinsically because Pearson's correlation is symmetric. And so you're making an equilibrium assumption without even meaning to. And things like this, to give you directed measures, are quite useful without being too complicated. So you can get directed measures from information theory, but there's a lot more you have to do in order to make those measurements. Okay. Yeah. The thing that jumped to mind for us that was the simplest thing was basically, let's just use the linear model. So linear models, as we've kind of heard in some of the other talks, they're obviously an approximation, but they're a pretty good approximation sometimes for fMRI data at least. And if you fit the linear model, you can fit it quite easily, and you can measure the entropy production exactly because we have the explicit solution for this. So there's this paper in 2024 from Nazari and Danny Bassett's lab again that I kind of used to justify this, which showed that linear models outperformed nonlinear models for fMRI data. So AR1, just one-step autoregression, outperformed the nonlinear models. As I'll show, it can basically be fit very, very simply. And it also implies fitting an effective network. So this matrix here can be thought of as an effective network of interactions between regions, which can then be analyzed with all of the network techniques that you're interested in. And you can kind of get interesting results out of that. It's also an asymmetric effective network. So it's quite difficult to get a directed network out of neuroimaging data in general. Other than time-like correlations, most of the other techniques are quite involved. And the last thing is that we don't assume that this process is stationary. If you fit this, you can actually look at the eigenvalues of B and check if it's stationary rather than just assume it from the get-go. And so this gives you a little bit of a kind of sanity check of whether what you're doing is valid for your data or not. Okay, so how can you fit this? I'll just show you the way we did it. So if you just discretize this process in time, you just discretize it forward with Euler-Maruyama, it's the simplest way to make this into a discrete time process. Get this, and this is actually completely equivalent to a first-order autoregressive process, AR1. And this can be fit with ordinary least squares or with convex optimization. And you can do all kinds of things with convex optimization. For example, you can say, I only want connections where I know that a physical connection exists, or I only want connections that have positive weight. And all of those constraints are convex, which means it's still easy to fit. There are some things that are difficult to fit. So, for example, if you want to say, I want B to be stable, so I want it to be a steady state, that's not convex. You can't do that. But as long as you keep the constraints convex, you can put all kinds of things into this, all kinds of assumptions, and it's still easy to fit. And then basically, it's very easy to recover from the AR1. You can recover your B and D matrix. So we did this, and we measured the entry production rate in task and resting state data. And then we also, I'm not going to go through all of the maths behind these measurements, but we measured the asymmetry of the matrix B. So the hypothesis was that entry production in neuroimaging data comes from asymmetry in effective interactions. So there's nothing to do with heat dissipation, nothing to do really with thermodynamics at all. It's just asymmetric interactions produce entry production statistically. And so we got the same result that they had in the literature with slight differences between tasks, that is higher in it's higher in tasks compared to resting state. But we also had this interaction network between regions that we could analyze. And so we measured the asymmetry with a bunch of different ways, and we showed that it was always higher. So the higher the asymmetry in the interaction network means the higher the entry production in general. And we showed this with numerical simulations as well, and with some analytical cases where this is just true. More asymmetry means more entry production. Okay. Once you have that network, you can do all kinds of things that you can't do with symmetric networks. So, for example, you can rank nodes. So there are all these techniques for ranking things in tournaments where you have directed interactions, for example. And you can use those algorithms to rank nodes in your network. And so with all of our effective networks, we can start to rank our nodes and see basically what happens based on the Yo functional network. So where does the subnetworks basically move between tasks? So this was more of a physics paper. We didn't go too much in detail with the interpretation, but you can see some networks like dorsal attention go to higher ranks or climb up the hierarchy in some tasks versus resting state, and others will go down the hierarchy in tasks compared to resting state. And it basically opens up this whole new avenue for kind of network techniques based on asymmetry rather than symmetric networks. So we weren't the only ones who had this idea. Like I said, Mathieu Gilson's lab did this with sleep data. And again, they're interested in the asymmetry in this connectivity matrix that you fit. And also Benozo's et al. Did this similar thing. And they looked at like how much is being sent from each of the Yo functional networks and how much is being received. And you have this new perspective that basically was, I think, missing on kind of, it's somewhat causal and it's somewhat asymmetric. And that it's not just about correlation. It goes beyond correlation. Another simple technique that I think is quite visually appealing and that we used in this other paper is network analysis of time series. So if I just told you if I gave you a time series and told you to figure out if it was reversible or not, it's actually quite difficult. It's not obvious. And there aren't that many strong techniques to do so, but this is one that was relatively recent and is really quite elegant. So what you do is you turn a time series into a network. And the way you do it is you basically do it based on this convexity criterion, which is called the visibility criterion. So I draw an arrow between points if I can draw the arrow without crossing an intermediate point. So if I can't draw an arrow between here and here because I've crossed this one, right? And it seems a bit artificial, but it comes from computational geometry. You're just trying to see what's visible from what. And this turns into a network. So each point is a time point, and the connections are between ones that are visible. Okay. If you flipped this time series around, all of your ingoing connections would become outgoing connections, and all of your outgoing connections would become ingoing connections. So if your time series was statistically reversible, the in-degree distribution and the out-degree distribution should coincide for an infinitely long time series. So a proxy measure of the irreversibility is to basically measure the divergence between the in and the out-degree distribution. Really simple, but quite elegant, I think, as a way of figuring out the irreversibility of a time series. So we did this in the multivariate case. So we kind of generalized it to multivariate dynamics. So what happens in the multivariate case is you have, say, two variables over time, x1 and x2. Each one becomes a layer in a multi-layer network. So you do the same thing for each variable alone, and you have a multi-layer graph. Okay. Then what we said is we want to figure out the irreversibility of a particular interaction. So what's the irreversibility between these three or these four regions in the brain? And so you basically make a joint in and a joint out degree distribution, and you measure the divergence between the two. And so the reason we did this was that this generalizes across scales. So I can do this for threes, I can do this for fours, etc. In a way that, say, pairwise correlations don't generalize. You can't do that for three. You can't do that for four. And so we were interested in trying to identify higher order interactions, which is a thing of kind of new interest in network science. Okay. So I applied this to a data set that was collected in Denmark by my friend Leonardo, who's at the Center for Music in the Brain. And he had quite a complicated paradigm, but we only just used a simple case. He had people listening to music in an MEG scanner, and he had it parcellating to these 6 brain regions that he knew were associated, or he'd shown were associated with auditory function. So you have the auditory cortex on the left and the right. You have the hippocampus on the left and the right. You have the medial cingulate gyrus, and you have the ventromedial prefrontal cortex. So 2 medial regions, a left side and a right side. And what we did was we measured the irreversibility of every possible interaction in the system. Because there's only 6 regions, this was combinatorially possible. You can't do this for 100 regions, but for 6, we could do everything. So we started off with just individual regions. So this is the irreversibility, where darker colors means more irreversible. And the star means number of standard deviations above the mean, and the dagger means below. So we showed that so at the first level, so just single regions, you find that the higher irreversibility is in your processing regions, and the lower irreversibility is in your sensory regions. And then in the middle, it's the medial regions. Okay, but we go through all the levels of interaction. So then you have your pairs. So you have all the possible pairs in your system. And we find that the ones that are highly irreversible are the ones that are a hemispheric pair. So this was kind of a thing that Leonardo said was expected if we didn't see this and that the method was not working. But we find that the highly irreversible ones are the pair on the right side and the pair on the left side. And the least irreversible one is the 2 auditory cortices. So this means that it's kind of different from a correlation, because you might have a high correlation between these 2 regions, but you wouldn't expect there to be kind of a significant interaction that was used in processing. But you see this hemispheric symmetry, and you see regions working together that you'd expect to. Okay. Then we go to triplets. Triplets is the worst combinatorially, so there's the most number of triplets, according to Pascal's triangle. And you find that the most significantly irreversible triplets are the ones that have the pair on one side and the ventromedial prefrontal cortex. So again, you see hemispheric symmetry between the left and the right side of the brain. Okay. The last 2, you have all possible sets of 4 and all possible sets of 5. So with the all possible sets of 4, we find that the highly irreversible ones are the pair on one side and the 2 medial regions, and the pair on the other side and the 2 medial regions. So again, you see hemispheric symmetry, and it makes sense that this would be the foursomes that were working together the most. And kind of an interesting result is this one here. The least irreversible of all the foursomes is the 2 most irreversible pairs. So this is kind of important whenever you're doing higher order things. You don't want it to just be reducible to the lower order things. You don't want to just be adding up all the pairs to make triplets and quadruplets. But if you take the most irreversible pairs, they don't actually work together as a foursome because you're missing the medial regions in between the 2. Okay. And the last one is the 5th order, where you can kind of think it as which region is left out rather than working as a 5-some. And we find that when you leave out the prefrontal cortex or the other medial region, you have the least irreversible one. So it suggests that medial regions are somehow mediating or orchestrating these interactions. Right. So another simple technique, not from me, that you can use. This is for spike train data because I wanted to cover scales like everybody else was doing. So for spike train data, you can't apply the techniques that I just showed you, but you can infer the kinetic Ising model itself. So this is a model-based approach, but as I mentioned before, it's the least structured model. So if you're going to use a model, this is the most justified one. Well, without going into biophysics, it's the most statistically justified one. And so what you do is actually very simple. You can either do it with likelihood-based inference, or you can do it with a mean field approximation. And there's a bunch of different mean field approximations from statistical physics for this. But it's quite simple. So you basically want to reconstruct the connections and the internal currents. And there are these really simple learning rules given from the likelihood. You just update your connections according to this rule, and very, very quickly you can reconstruct the connectivity matrix. And I tried these methods myself, like with simulated data, and they work really well. They work really simply, really quickly as well. And so, yeah, you reconstruct the connectivity matrix and the internal currents. Slightly different because this gives you some positive valued edges and some negative ones, which mean inhibition and excitation, I guess. But there are much fewer techniques in network science for studying things with signed edges. It's quite difficult to really interpret what a signed edge means outside of neuroscience. So now I'm going to turn to the outlook and to the how much time do I have left?
There is lunch now, so
okay.
You can take as much as.
No, I won't take too much of your lunch time. Okay. I'll be quick. Okay. So everything I've talked about is kind of analysis of neuroimaging data, and not about computation in neurons. But I am interested in computation in neurons. So what about non-equilibrium physics can we use to kind of build models of computation? So some of the classic models of computation rely on equilibrium assumptions. So the discrete Hopfield model with stochastic dynamics is just a symmetric Ising model, and so it's reversible. The continuous Hopfield model is symmetric. It requires this symmetry of connectivities, and that means that it defines a gradient flow or a generalized gradient flow. So what Hopfield showed, and he used it as a feature, not a bug, is that recurrent neural networks with symmetric connections are they're defined by an energy function, and you just flow to the minimum points of the energy function. As I'll show in the next slide, he used this to build models of associative memory, as we know, but it prevents all kinds of non-equilibrium behavior. You can't have limit cycles, you can't have chaos, you can't have anything if it's just an energy function. Okay. So yeah, this is what Hopfield did. He took this very simple model. This is the continuous version of the model from 84. Then you have the discrete one from 82, which is probably more well known. And he basically showed that this is just, if it's symmetric, you have an energy function, and you're just going to go to the minimum points of the energy function. There's nothing else you can do. Now, of course, you can use this to recover memory. So this is 32 by 32 network of neurons fully connected, and I trained it to encode this picture of a cat dressed as a shark. And yeah, all you're going to do is go to these minimum points, and it will recover memories, but it can't learn sequences. And so we wanted to develop techniques to train RNNs to learn sequences based on the asymmetry of the connectivity, using the asymmetry of the connectivity. Now, doing so in a biologically inspired way is still an open problem, and it's quite difficult. But we came up with a method that isn't biologically inspired. And I might have to go a bit quickly this last bit. So the idea that we had was that we don't need to figure out how to do dynamics in the high-dimensional space, because there's a bunch of different evidence that suggests that neural dynamics are constrained to low-dimensional manifolds, and we're just going to do dynamics on a low-dimensional manifold rather than in high-dimensional space, and this is going to make life easier. Okay. So what we did was we say, okay, we're going to have a recurrent neural network just like Hopfield did, and we're going to make sure that it only lives on one subspace. And in general, the manifolds are nonlinear, but we're going to just do things on an affine linear subspace to make life easier. And instead of doing what people usually do with neural manifolds, which is like reconstruct it from data, we're going to do the opposite. We're going to design a recurrent neural network that has particular dynamics on the neural manifold. We want to specify what the dynamics are. So we want them to follow some target process, and we want to train it. So we want to fix the weights, the input biases, the noise, and the manifold itself to follow this target process. And we came up with a technique to do this called drift diffusion matching. And it's totally non-biological. But what we do is we basically say, what are the dynamics on the subspace given our network? And we're going to assume that the network has a low-rank structure. So the low-rank structure means that you're constrained to this manifold itself. So I'm going to skip through some of the maths here so that we can go to lunch. But yeah, so we're assuming it's low rank. We're assuming it lives on this manifold. Well, we're making sure it lives on this manifold. And then what happens is your recurrent neural network becomes equivalent to a two-layer perceptron. And you can follow the maths for this, but you'll see that basically the dynamics on this subspace become parameterized by a two-layer perceptron. And so what you have to do to approximate any target vector field you want in the subspace with your neural network is just train a two-layer perceptron to approximate it, and then use the parameters to reconstruct your recurrent neural network. And then you'll have an autonomous circuit that can encode whatever dynamics you want. So you can train a recurrent neural network that when you sample from it, it will produce dynamics that look like this. And when you look in the subspace that you learned, you'll see whatever you put in there. So you can train it to do van der Pol oscillations. You can train it to represent the Lorenz attractor, the de Grasse attractor in this latent space. And of course, you can reconstruct it from the trajectories as well. If you do PCA, because this is a linear subspace, you'll reconstruct the thing that you put in there. So here, you won't reconstruct it exactly, you reconstruct it up to a transformation. So if I train it to do the Lorentz attractor, and then I do the PCA, I'll get something that looks like a shifted Lorentz attractor. All right, so I don't have time to talk about this, I guess, but we use this to develop not a very biological, but a bit mathematical model of input-driven associative memory. So we added some input to the system, and we created a system that could basically switch between memories based on the input signal. And the analogy that Alain likes to use, my supervisor, is that it's basically we're tilting the potential in space so that the ball, based on the input, rolls, the ball would be the neural activity of the neural manifold, rolls into the correct memory. And so there's some kind of mathematical reasoning for how this works that I'm going to skip. But this is effectively what we're doing. So you start with a system that has 4 memories in your neural manifold, which is just minima of an energy function. And then based on the input, we're going to basically tilt the board so that the system rolls into the corresponding memory. And so you can build examples like this, where you have your neural network, and then you have your input signal. So first, the input is set to, you can pick however you want to code your inputs. You set it to memory 1, halfway through the trajectory, I switch it to memory 2, and you'll see the network kind of reacts to this change in input. And if you look on the neural manifold, you'll see we converge to memory 1, and then we converge to memory 2. And you can do this with whatever coding scheme you want. So we tried one hot, we used random Gaussian signals, etc. Okay, so let's look at this. Yeah, so irreversible dynamics are not just interesting in neuroscience, they've also become state-of-the-art in some machine learning disciplines. So irreversible stochastic processes are the foundations of generative models, diffusion models. So these work by basically taking a stochastic process that goes from some point in your data set to noise and then reversing it. So you basically go from noise to new samples from your data set. It's being used to develop new versions of flow matching that take into account non-equilibrium things, take into account basically currents in your system. And so it's a bit of kind of mathematics and physics that is having new applications in all kinds of areas. All right, so some conclusions. Statistical methods for neuroscience are overwhelmingly based on equilibrium models, and we have kind of shown now that this is inconsistent with neural data. So it's an assumption that might be grievous or not, depending on the exact case. So switching to a non-equilibrium paradigm means basically embracing asymmetry or developing methods that focus on the asymmetry between connections and the time irreversibility of the dynamics. Measuring irreversibility and asymmetry is hard, but there are some techniques that allow you to do this. And it has implications for both models of neural computation and, in general, for advancing generative machine learning as well. So thanks a lot. If you're interested in the work, we wrote this review paper, which is kind of most of what this talk was about. And then there's also a number of more particular studies, results-based studies. So thanks a lot.